Prokaryotic cells do transcription and translation at the same time. When a protein is no longer needed, transcription just stops, and therefore everything else stops. So that's kind of like, it seems nice and simple. Eukaryotic regulation involves transcription, but it also involves translation, just like prokaryotic cells, but the two processes they're physically separated, and a lot can happen between transcription and translation because they're in different places of the cell. So there are different chemicals and regulators for the processes. Let's start with prokaryotic cells. Recall that they have one large supercoiled circular chromosome that's in the nucleoid region. And somewhere on the chromosome is the tryptophan operon, it's just a collection of genes in a particular location that control the synthesis of one thing. And here we have tryptophan. In an operon, there are regions, and unfortunately, different books call them different things. It's just a teaching concept. But this is in general how it goes. First, you have a sequence, and I don't care what you call it, but it's a promoter, generally. The promoter is where RNA polymerase comes over and starts to bully its way into the DNA and start transcription. Okay, and then we have the operator region, and this is a place where a repressor protein can bind and prevent transcription. It sits there and it blocks RNA polymerase from getting any further. And then lastly, just after the operator is the coding sequence. It's the actual first codon for the protein. Sometimes, like your book calls this the transcriptional start site. I don't care whatever you call it. It's the start, and it leads to the region of the gene that actually contains the recipe for the amino acids. All right, what does this all mean for you? It means that we have a place for RNA to come in and bind to. And this place says, here, bind here. That's the promoter. But right next to the promoter, we have a sequence on the gene that RNA polymerase then has to get through and actually make the promoter, the protein, and that's called the operator. It's like the accelerator and brakes. You have to turn one on and you got to turn one off. What you want to take away from this is that prokaryotic cells regulate protein synthesis by regulating transcription. It either happens or it doesn't. And what determines that is the promoter and the operator. Let's talk about eukaryotic regulation. And for a moment, let's think about all of the chromosomes, right, in a human 46. Recall that your chromosomes have those nucleosomes with the 4 histone proteins and the DNA that's wrapped around them as pictured on the left. This packed or coiled the DNA into their condensed forms of chromosomes. Nucleosomes can slide along the DNA, and when the nucleosomes are really close together, they form a barrier, and RNA polymerase just can't get in there to start transcription. And when the nucleosomes slide apart, as in the picture on the bottom right, they allow RNA polymerase to get in there and start transcription. There are chemicals, right? There's chemicals everywhere. There's chemicals just kind of floating around in the nucleus that can bind to these nucleosomes, and they can make them cluster or spread apart. So there's a process called methylation that adds a methyl group to the chromatin, and it clusters the nucleosomes together. But adding acetyl groups will spread them all apart. Is this what's happening in people who have autoimmune diseases or cancer? Which cancer can be considered autoimmune? Are genes that shouldn't be activated being activated? Or are genes that should be on getting turned off? This is what we call the epigenetic level of regulation. It's a very large scale level, and it can turn on and off entire chromosomes. Female cats with two X chromosomes will turn off one of the X chromosomes. Hey, you don't need it. Why not? So just package it up and leave it over there. So this is a very large scale level and can turn on and off entire chromosomes. That's epigenetic regulation. The transcriptional level of regulation is a little bit more specific than those large-scale nucleosomes moving about, but in general, in this level, we are just determining if transcription can even start or stop. This is kind of like the type of regulation in prokaryotes, right? We're just saying RNA polymerase, go ahead, do your job or not. Eukaryotic genes, they also have something that we could call a promoter region. It's a bit more complicated, but let's just keep it at that for now. The promoter region can interact with basal transcription factors. What is that? It's just a chemical in the cell. So basal transcription factors will bind to the promoter, and they will encourage transcription. Make more, make more, make more. They only encourage. There are other factors that are called specific transcription factors, and they're much more complicated, as the name indicates. They interact with other regions of the DNA. You want to know something? The main point to remember is that specific transcription factors enhance or repress transcription. Recall that the mRNA has to travel from the nucleus to the cytoplasm between transcription and translation. Also, the mRNA is spliced to remove introns and exons, and there are other modifications that take place before it leaves the nucleus. The enzymes that do the splicing are called spliceosomes. Really? I'm sorry. Remember that splicing can create many proteins from one gene, as pictured here on the left. And this is a process called alternative splicing, which is fascinating. And there's a thing in your textbook that leads to a video that's really kind of awesome. But alternative splicing wasn't discovered until the 1970s. There are other processes that can be controlled at this time. The mRNA receives a cap and a tail before leaving the nucleus. This is like put your boots and hat on before going out. These processes, they can be controlled too, and they can be controlled to add or not add the cap and the tail. Just like you might freeze outside in the winter if you go out without your boots and hat. The mRNA will decay at a faster rate once it leaves the nucleus. So all mRNA decays at some point. I mean, it is a disposable copy, but the cap and tail can regulate when that happens. The regulatory processes pictured here on this slide are ones that would be considered to control translation, either during it or maybe a little bit after it. Translation from the ribosome's point of view is extraordinarily complicated. Assembling the small and large ribosomal subunit is complex, and there are many ways in which that coming together can be regulated. Other processes focus on the proteins that bring tRNA to the ribosomes, like chaperones, and post-translational regulation. Once the protein is already made, that can involve the folding of a protein. In chapter 3, I mentioned, maybe, these proteins called intramolecular chaperones, and they're proteins that fold other proteins. These can be prevented from doing their job. This picture here is just a small subset of many of the ways in which translation and folding can be regulated in general. Don't forget, regulation is not always about stopping the protein from being made. It is also about encouraging or promoting or enhancing that the protein be made. In another class of mine, we do a really, really involved case study of cystic fibrosis. And we find out that the disease is a result of the malfunction of one protein in the cell membranes of cells in the respiratory tract. So that's the trachea and the lungs. This protein is obviously made from the instructions of a gene, and so this disease is genetic. At first, students think that, oh, the protein must not be made, right? And that's the disease, the protein's not made. But that's not exactly the case. Sometimes versions of cystic fibrosis, the protein is made, but it's the wrong shape. Sometimes it's made, but it gets inserted into the cell membrane at the wrong place. And yes, sometimes it is just not made. But finding a cure for this disease is explored by way of a gene or genetic solution, among other solutions. How can we find chemicals that will encourage this cell to make the protein, and then another person needs a solution to make the protein correctly, and then still another version of the disease needs the protein to be inserted into the cell membrane correctly? Cancer is a collection of diseases that includes tumor-producing cancers, and that's what we usually think of, but it also has blood cancers like lymphomas and leukemias, where there's an overproduction of cells or associated proteins like antibodies. It's like too much of a good thing. However, linking them all are the proteins made by genes that control the cell cycle. And we call the genes that control the cell cycle proto-oncogenes. These make proteins that interact at the cell cycle checkpoints. If these proteins stop working correctly, they are considered oncogenes at that point. Cells will continue to divide even though they have mistakes in their DNA. This is like, and I'm just thinking of cancer here in a general process, just general overview. I want you to think of cancer like there are genes that are accelerators. These genes stop functioning, and the accelerator pedal on the car gets stuck in the on position, and more cells are being made no matter what. Tumor suppressor genes, like the p53 gene, can stop the cell cycle from continuing out of control. It's like the brakes. So cancer doesn't result from one thing. It results from an accumulation of mutations in these types of genes. This rate of accumulation is, it's quite slow for most people, but as you age, you accumulate more and more mistakes in your DNA. And that's why the risk of cancer increases as you age, but it's also why more older people with cancer have it.